Sejam muito bem-vindos a mais um Pasteur Laria podcast, o podcast oficial da Spektra. E hoje aqui, nesse dia tão especial, não usual, porque não temos Natan e Maria. Temos Natan e Maria, Iris?
Não temos Natan e Maria.
Quem nós temos hoje, Iris?
E embora não seja usual, vai começar a ser um pouco mais normal esse cenário aqui também.
Esse é o novo normal, como diziam
exato.
na pandemia.
Exatamente. E hoje a gente tem dois consultores da Consult Lasa com a gente para discutir.
Os dois já bastante conhecidos de vocês, eu acho. Uma mais do que
uma mais do que outra. Uma já esteve aqui junto a nós.
Sentada nessa mesa.
Pertinho, inclusive muitas saudades dela.
Sim.
E o outro é um grande pesquisador, não só da Consult Lasa, mas da Lasa também.
E num contexto até fora da consultoria, se pensar na questão de autoria de paper, é lógico que
produção de ciência, né?
Produção de ciência mesmo. Então, eu já vou introduzir, nós vamos ter tanto a Ari quanto o Martin aqui, o Martin Chilton, convidados da Consult Lasa para essa conversa, que vai ser sobre o Qualification Paper, e nesse caso, pensando no Martin, a gente tem uma pessoa que é realmente uma referência que extrapola o contexto da Consult Lasa, justamente por suas publicações que são utilizadas em guias, como o guia ICH-Q3E, e realmente mostra a relevância do trabalho que está sendo feito por esses pesquisadores, né? Então, eu já dei o spoiler, obviamente, mas Iris, qual que é o tema de hoje?
Hoje, ou como vocês já viram na thumb, hoje a gente vai falar um pouquinho sobre a versão final do Reflection Paper de Qualificação de Impurezas Não Mutagênicas da EMA, em que são discutidas realmente todas as abordagens que podem ser utilizadas para avaliação da toxicidade de uma impureza quando ela extrapola o limite de qualificação, e a partir de então garantir a segurança da exposição a ela. Eu acho que isso é muito, muito importante. Saiu agora realmente a versão final, mas a gente já tinha uma versão draft que saiu no início de 2025, se eu não estou enganada, e uma anterior que já é de 2019, que também já trazia um pouco sobre essas abordagens alternativas, que é o futuro mesmo, né, de abordagens alternativas ao uso de animal. E que eu acho que é muito importante realmente, agora que finalizou, que não está mais na versão draft, né, está oficializado, realmente demonstra o quanto que a EMA está preocupada com esse tema e mostra a importância de uma discussão toxicológica via NAM, né, essas New Approaches Methodologies, né, que é em métodos alternativos ao uso de animais. E eu fiquei muito feliz que saiu a versão final, porque eu esperaria ficar
tão aguardada.
Ficaria, porque como ficou em draft de 2019, depois fez um outro draft em cima do draft, e agora fiquei muito feliz que saiu essa versão final, e ela conversa inclusive com o próprio guia que agora a Anvisa publicou recentemente.
Era exatamente isso que eu ia falar, sim.
Então, fala aí um pouquinho.
A Anvisa, assim, eu não sei até que ponto isso é uma coincidência do destino, ou se isso foi acordado de alguma forma. Imagino que não, mas a Anvisa recentemente também publicou um guia sobre a qualificação de impurezas, que é o guia 83. 83? Isso. É o guia 83 que foi publicado, acho que em dezembro do ano passado.
Putz, é bem recente.
Eu não lembro se ele é do final de 2000.
Ele está em consulta pública.
Está em consulta pública, exato.
Importante.
Mas ele foi publicado ou agora no finalzinho de 2025 ou no comecinho de 2026. Bom, estamos no comecinho de 2026, então agora, recentemente. De dezembro de 2025, certo?
Dezembro de 2025 foi publicado e temos até julho de 2026 para contribuição.
Para contribuição na consulta.
Então, eu acho que é muito interessante, inclusive, esse episódio, para que vocês tenham, né, já uma familiaridade com o tema, porque no Guia 83 ele traz muitos aspectos que a EMA descreve no Reflection Paper de Qualificação de Impureza Não Mutagênica, e agora que a gente vai discutir sobre esse tema, acho que também é importante para que a gente consiga ler com maior direcionamento o Guia 83 e, inclusive, propor nossas contribuições que a Anvisa está esperando que a gente faça.
E dessa forma, a gente pode até harmonizar, de certa forma, os guias e harmonizar as discussões para que realmente o que seja publicado finalmente no guia, com a publicação no guia, esteja de acordo com as normas internacionais, que já está, inclusive, a gente já discutiu o Guia 83 aqui internamente bastante, e quem sabe, se vocês quiserem, não possa ser um episódio futuro para vocês,
Sim, com certeza.
comentário sobre esse guia. Então, coloquem comentários aí se vocês pensam que essa discussão é uma discussão de interesse para vocês. Eu espero que seja. Mas realmente, então, mostra como há uma harmonização dos conceitos e como há uma harmonização das discussões sobre a qualificação de impurezas.
Sim.
E
Eu acho que a gente pode começar. Certo?
Podemos começar, então vamos passar para os nossos convidados. Acho que vocês já cansaram da gente discutindo sem chegar no momento de verdade.
Só fica no...
Só fica
aguarde.
Não, espere, vamos conversar sobre isso. Não. Mas então agora, nós vamos passar por inglês um pouquinho. Então, se você tem um pouco de dificuldade com o inglês, eu imagino que existam várias ferramentas já disponíveis agora para...
Antes de você passar por inglês...
Eu sei, eu posso fazer essa função.
Vai.
Antes de passar por inglês, a gente quer soltar também os recados.
Os recadinhos, Leandro.
Os clássicos recados.
Que
temos no nosso podcast.
Ó, Thaíris. Fala aqui, ó, do nosso parceiro, o Sergião, a Axios Research, que o Sérgio também é representante da Simple Systems. Se você quiser padrão, você vai lá com a Axios, falar com o Sérgio. Sérgio, quero padrão, vim no Pasteur Laria, quero 50% de desconto e 90 dias para pagar. E não vai te dar isso. Pede uma caneca, um brinde.
Ou um ursinho.
Ou um ursinho. Ursinho eu não garanto. Pede um brinde. Eu garanto 90 dias de prazo de pagamento do que o ursinho. Mas
o ursinho é especial. Nossa, essa é a minha grande pergunta.
Eu não vi esse ursinho em lugar nenhum. E olha que eu fui com o Sérgio para Action lá no Canadá. E não vi esse ursinho em lugar nenhum. Embora que o ursinho é da Sampling System, né?
É verdade.
Eu não fui para Sampling System ainda. Sérgio! Então vai lá, fala com o Sérgio, pede padrões, pede forma de pagamento, pede tudo, faz um combo entre padrão e amostradores que a Simple System tem, amostradores de inox, viscoso, pó, amostrador que você quiser tem na Simple System. Beleza? Vai lá com o Sérgio, fala que você viu no Pasteur, que é importante, pede as coisas tudo para ele e ajuda nós, que ele vende e a gente ganha dinheiro aqui no patrocínio e o podcast continua. Meu salário depende disso. Você vai lá com a YMC, tá lá atrás da Iris lá agora, ó. YMC Research, mentira, YMC America. Fala que pede coluna, vai falar com o Laudo, vai falar com o Lucas, vai falar com a Marina, vai receber treinamento, vai ver se o equipamento está tudo certo, sua coluna tá boa, tá quebrando, não tá. Fala com eles que eles têm tudo para te atender e o Laudo tem a McLaren das colunas, que é a Tri-Art, que é a coluna mais cara do mundo, segundo eles. Tem mais cara?
Eu não conheço de carro e nem de coluna.
Eu não busquei o preço de colunas recentemente pra te dizer.
Mas é da lenda que é a Dolce & Gabbana das colunas. Agora, assim, a Dolce & Gabbana é tão chique assim, mas é a coluna das colunas. É isso que tem. Você vai lá, fala com eles, fala que você viu aqui, que eles vão te dar desconto, treinamento. Aí você faz o pacotão. Compra a coluna lá, compra o padrão com o Sérgio, ponta pra rodar no seu equipamento HPLC e vai ser feliz.
Uhum.
E lembra pra gente, segue a gente nas redes sociais. Podcast Pastelaria Oficial no YouTube, podcast Pastelaria Oficial no Spotify, dá 5 estrelas pra gente, avalia a gente, curte, compartilha isso aqui, que é muito importante a gente chegar na meta dos 3.000 inscritos no YouTube, porque o Spotify tá passando no YouTube, hein? Então ajuda nós a atingir a meta do YouTube, que é a meta do ano passado ainda. Vai lá, ajuda a gente, tem o nosso site também, você vai no nosso site lá, tem tudo que você precisa, tem playlist dos episódios do YouTube separado por categorias, então se sua empresa não deixa você ver o YouTube, vai no site que você consegue ver. Não consigo ver o site, vai no Instagram, porque tem um monte de coisa no Instagram também, no Spektra Soluções Científicas, não consigo ver o Instagram, mas sua empresa deixa você ver o LinkedIn, então vai lá no LinkedIn, Spektra Soluções Científicas, veja tudo bonitinho, segue a gente, compartilha e espalhe a palavra, tá bom? Um beijo pra vocês, bom programa, vai pro inglês agora.
Não, mas antes de ir pro inglês, eu só quero dizer que a cada episódio que passa, o Leandro me surpreende com coisas que ele aprendeu de amostragem e coisas do tipo. Eu acho que a gente pode dar um relatório pra ele.
Eu sei teoria, não sei pegar na prática.
Ele fica falando que o salário dele depende dos patrocinadores, mas agora ele pode...
Ele já consegue escrever um relatório, né?
Eu sei na teoria. Prática, o que isso quer dizer? Eu não sei, eu sei o que faz e a junção.
Você sabe pra que serve uma coluna, Lê?
Pra filtrar alguma coisa.
Tá quase lá. Talvez mais uns...
É pra separar. Separar é uma melhor palavra.
Mais uns 10 episódios de podcast ele tá pronto.
É que tem que falar sobre coluna, não teve episódio sobre coluna. Não, teve sobre a HPLC.
Sim.
Na coluna, especificamente, qual que é a função da coluna, ainda não.
É o coração da HPLC.
E do equipamento de cromatografia.
Isso, qualquer cromatógrafo.
Que a HPLC é a técnica, né?
É uma cromatografia líquida, É o equipamento que faz cromatografia líquida. E líquida porque como a gente vai... É uma técnica de separação, de certa forma. Em que você tem uma amostra cheia de componentes e você quer separar esses componentes para conseguir identificá-los. Identificá-lo, quantificá-lo, daí aí o verso é grande. E essa mistura passa por essa coluna e a depender do solvente que você coloca, extrator, junto com a matriz, junto com as características dos componentes que tem nessa amostra, ele vai separando para você e vai meio que filtrando agora sim, pouco por pouco os compostos. Então, no final das contas, ele separa os compostos que estavam numa matriz única em agora diferentes piquinhos exclusivos para cada composto. E assim a gente consegue avaliar, por exemplo, se o seu composto, sua impureza não mutagênica está acima do limite ou não, por exemplo.
Resumindo, gente, vai nos 3 mil inscritos lá, me ajuda.
Ajuda, André.
Bom programa para você.
Bom, mas agora, passando para o inglês, vamos aos nossos convidados de hoje e vamos a esse episódio tão especial aí. So, I guess today we have 2 very special guests here. We have both Ari and Martin from Consult Laza, and they are today with us for us to discuss the non-draft anymore, but the qualification, the reflection paper on the qualification of non-mutagenic impurities. So after, I don't know how many years, but more than we would like, we finally have the final document for this. And I suppose it's a very important mark that we have now, because now we have finally a published guidance on the qualification of impurities that I think it's a very important topic, especially when we are considering the industry. And we finally have this guideline that gives us some pathways for us to try to qualify an impurity, because this is a topic that doesn't have it's a very sophisticated discussion, because you can use several different
approaches
to qualify an impurity. So here we can maybe identify which approaches we can use and finally guarantee that the impurity will be safe in the levels that it's being qualified. So I think we can start with when should an impurity be qualified? I think this is the most important question. What is the qualification of an impurity that you can talk about? Maybe Martin, what's your experience on this? What is the qualification of an impurity?
Yeah, great question. Hi everyone, nice to be with you. Thanks for having me. Yeah, I guess basically, I guess one of the things to say is we've still got ICH guidelines that are relevant here. So Q3A, Q3B, and they will typically tell you what levels of impurity you can have in your drug product, depending on the maximum daily dose and other factors. So usually we will want to qualify an impurity if we have an amount above one of the qualification thresholds that's set in Q3A or Q3B.
Yeah. And by qualification, we mean by assessing its toxicity, because as the value of the impurity on the drug product or the API, it's higher than what the regulators expect to have. We need to assess its toxicity in the amount that it's being observed on the product in order to guarantee safety for the product and the impurity itself also. So it's really important to do that.
Yeah, and a question for Ari now, because I believe that you have a lot of experience in the industry with all the aspects on the qualification and degradation products. And when do we need to qualify something? I mean, by an industry standpoint, when does this happen? When this necessity comes?
Yeah, good question. Hi guys. Nice to see you again. But I'm now in the other position. Yes, I was at Spektra, but now at Consult Lausa. It's a good question because, for example, when we are in a pharmaceutical industries, thinking about, for example, generic industries and also big pharmas, they all develop stability studies. So when we are developing a new product, even a new API, you should be concerned about the levels of these potential impurities that can be formed, since the route of synthesis or during your stability studies in your final product as well. And it is interesting to see because if the limit is above of the qualification thresholds, this is the as Martin said before, you should qualify these impurities, but where we can measure this is just because we have the stability studies. And this is important because now in this reflection paper, EMA, they are so clear regarding, we should qualify and evaluate the toxicity of your impurity based on the batch of your studies. So which kind of batch they are talking about here? Of course, this is stability studies. They don't want to receive any kind of documentations, qualifying periods so far away from the limits that you are seeing in your stability studies. This is a really good gap that now it's clear in this reflection paper.
Mm-hmm.
Yeah, and I think this is also pretty in line with what Anvisa expects from a qualification, from the qualification of an impurity, because they never want us to establish a maximum limit for the compound. They always want us to evaluate the toxicity of the impurity in the amount that it's being observed in the stability studies or on other studies. So they really expect us to evaluate what we see, not what we can.
The real.
Yeah.
And for me,
the real amount.
It's something that's not being discussed on the reflection paper, but it's something for me that is really close to it, is as we need to qualify the amount that's being formed, that's being seen on the product, the ways that we establish its the stability of a product, it's beyond the like in the opposite way of it, because till now we do some tendency analysis to maybe see what we expect, what is the amount that we expect to be formed to qualify that limit, and then when we say when we are in the end of the stability studies, we see that oh no, we qualified higher than we needed because we made some expectation of it based on the tendency that is not going to be real. So this is something for us, maybe for generic industries, it's like a more important problem to discuss, because we tend to do a lot of tendency analysis in order to expect the amount of the impurity that is going to be formed, but at the end of the study we see less. I hope so.
Yeah, I have.
Hopefully,
Yeah, I have a point here, because as I know what Anvisa expects for, and also having the opportunity to see what the other agencies around the world they are expecting to. There is it's funny because in Brazil we can see most of people asking for like, oh, how is the highest amount that you can qualify for me this impurity?
Mm-hmm.
And this difference from our side here, out of Brazil, because people came with the final limit, like I need these limits because I'm seeing this in my stability studies. So I don't know what's the trick of the timelines they have, to work with the different kind of agencies. But it's nice to see, like, okay, it's I don't know if it's culture, like to Brazil ask for like, oh, what is the maximum limit that you can achieve, you can qualify to me this impurity, and here we can say like, no, I want like 1%, 2%, whatever, how much they need it,
Yeah, Chacha, about that, I believe maybe I'm wrong, but there's some huge difference between us both because we are in the different climate zone. So maybe
Yes.
it's more difficult to make the product stable because we are on zone 4B. So
They are anticipating.
Yeah.
So the companies who is in the worst scenario regarding the stability studies, they are anticipating to like,
Yeah.
okay, qualify these limits, the maximum that you can qualify. But in the end, okay, if it didn't achieve these limits, we can review the documents before submit to authorities. Yeah.
After we establish when or the amount that we need to qualify, and after understanding when we should qualify an impurity, we can now start to evaluate the toxicity of the compounds that you are trying to qualify. And I think it's really important that you know the structure of the compound. This is something really important because we sometimes have some issues with this. We cannot qualify something we don't know the structure because all the toxicological discussions, they are based on what we see and what we can infer based on structural alerts, for example, or maybe on with the comparison of the compound with a surrogate, for example. We need to have an established structure for the compound for it to be qualified. And the reflection paper here, it talks about the qualification of non-mutagenic impurities. We also must understand if the impurity is mutagenic or not. And for this, we typically can use in silico tools or an Ames test, for example, to understand if it's mutagenic or not. Ideally, here we understand that in silico tools are quite powerful for this, to evaluate if the impurity is mutagenic or not. And we can do this using at least LASA softwares like Nexus with Derek and Sarah. And we can establish these according to the ICH M7 classification.
Yeah, and something about that on the reflection paper, they say to us about we have like a legislation that talks about using QSRR to establish mutagenicity of impurity, that it is being like established and being well assessed. But now they started to discuss that you actually can use this kind of tools to evaluate a non-mutagenic impurity as well. So they know that. So for me, what is more like the most important thing about this reflection paper, that we can harmonize the ways that we can assess the toxicity of impurity and like to be comfortable in the approaches that we want to use based on the data that we have. And now we can we can dialogue better with the agencies as well. So because everybody is harmonized and everybody is following this guideline. So for me, this is really important.
Yeah.
At the time that we are now.
Yeah. And so after we have all the cards on the table, so we have a structure, we know that it's not a mutagenic impurity, then we can really look into the different strategies that the reflection papers brings for us to try to assess the risk of the impurity, the toxicological risk of the impurity. And we also have strategies to try to qualify the toxicity aspects of the compound of the impurity that we are talking about. So I think the first strategy that is brought on the reflection paper is the strategy to qualify via the metabolite route. So here it establishes that if the impurity is a relevant metabolite, then it's can be considered reasonably safe. Yeah, and I think this is quite different. Actually, this in this topic here, we have a lot more information when we compare to the previous version of the reflection paper, of the draft version. So, yeah, what do we have here?
Yeah, and I have a question for Ari about it, because I know that she had worked a lot with qualification of non-mutagenic impurities before the previous draft has come up.
Yeah.
So I want to know, Chacha, from your perspective, what has changed about because the metabolite discussion has been one of our opportunities to assess toxicity since the beginning, since the first version of the draft on 2019. But now, as Lucas said, it's being more robust. What is like the approaches that we must evaluate for assess the impurity as a metabolite? So I want to know from you, like, if you can tell us a little bit of your story about it, about the beginning of it.
Yeah, it's interesting because, for example, I did most of these assessments from Brazil perspective, and I know that we had in the past a guidance by Anvisa that they are clear. They are clear explaining, like, if something is, what is a metabolite? Is anything, it's a compound who is more than 10% into your body. So something that converts in 10% in your body. So we did a lot of this discussion in the past, explaining this kind of strategy. So if you have, we can discuss, for example, dipirona, metamizole in English, sorry. Metamizole. Metamizole, this compound form another compound by hydrolysis, and this, because metamizole is a pro-drug, so they form the main compound who will have like a pharmacological effects in your body. So this is why, and it will be forming more than 10%. So this correlation will be a pharmacokinetic, is a pharmacological effects. It's more than 10%. So this is considered a metabolite. But now it's different because they bring this more, they are clear. They bring, this is not the ideal how to consider something as a metabolite, because sometimes we have chemical, how can I say, chemical effects, for example, hydrolysis, oxidation, that there is no biochemical biotransformation. So it's more a chemistry transformation because of pH or whatever. So this is important to how to measure. And here now in this reflection paper, they are more openly to discuss, we will consider this compound as a metabolite based on this and that. And they are showing more math behind all this, how to calculate this. And this is so different that we have like many years ago.
And it gives like the opportunity for us to qualify an impurity based on the metabolite discussion more often than before, because before we only had the discussion about the metabolite as the amount was being formed as for at least 10%. But now we can discuss based on this math, based on the paper from Weidorf. We have the opportunity to discuss the metabolite formation like more times,
I would like to add here that, for example, it's not a merchant, but it's important to mention that we also have in-house the Meteor. Yeah, we can use Meteor as an in silico tool to discuss potential metabolism for these impurities, if it's real or it makes sense to be formed from the API. So yeah, it's another great opportunity to discuss, because it's one of the tools that we have to bring expert review, expert discussions into our assessment.
And one thing that like in comparison with the draft version of it, and something that got Lucas really worried about it.
Yeah.
Yeah.
Before the recording, we had a brief chat, and I was like, "Oh my God, we cannot use this anymore."
Yeah, because now to assess if impurity is a metabolite, also we need to calculate the ratio between the amount being formed as a metabolite by the amount that we expect to like the dosage of the impurity. And if the compound is still toxic, so you see when I'm where I want to go by. This ratio needs to be higher than just like assessing that the amount of the impurity being formed is higher, just like a little bit higher than the impurity. And for me, this is really important because it states that we need to assess the toxicity on like a very,
a general
approach.
Yeah,
you just like cannot see the metabolite and say, okay, so the impurity is also metabolite, so that's it. For me, it says really, it sticks to the fact that you must assess the toxicity on the general background. So you need also to understand what are the effects of this metabolite, also an impurity, to confirm how much of this difference between the metabolite and the impurity it's like okay to have on the product, so we can ensure its safety. So for me, it's not only being assessed on the metabolite aspect, but during the reflection paper, for me it's really clear that they want us to assess on the general aspect of the impurity and to maybe find the best approaches in order to guarantee its safety.
Yeah, and this basic toxicological assessment can also be done via in silico tools.
Yeah.
And Derek is a great tool for this. So it can look for structural alerts, relevant structural alerts, and thus we can identify if the impurity is supposed to be very toxic or supposed to be less toxic. And from this we can establish some safety factors that can be used on this kind of evaluation via metabolite of the impurity.
And by safety factors, we can pass through to read across.
Yeah, and also talking about in silico tools, because read across is the next topic, and it's also really related to in silico tools. And I think read across is one of the most powerful tools that we have available for establishing toxicological aspects of a drug, of a impurity,
based on new approach methodology.
Yeah, based on new approach methodology, because we rarely have information on the toxicological aspects of the impurity, because it can be a new molecule that is not it was not previously identified, and so it's really difficult to have accurate data on the compound that we are trying to qualify. And so we can compare this molecule that we have with other molecules that we know the toxicological aspects of, and this strategy is called read across, and it's a good tool to fill gaps that we identify on this toxicological evaluation that we are doing. And I know that read across is also really tied to ENL evaluations, because usually when we see extractables and leachables evaluations, we don't have data on the toxicological aspects of the leachable.
Because chemistry, it's really mad, everything can be formed.
and it's really interesting because I think ENL they are not in the usual chemical space of the drugs, and this is something that that makes the toxicological evaluation even harder because it's more difficult to find data on the ENL. So I don't know, Martin, do you have some comments on this? Because I know that you're one of the specialists of ENL. So how do you compare this read-across evaluation of an ENL to a comparing to the qualification of an impurity?
I think there's a lot of overlap, personally, between the two. Like you say, the main difference is the chemical space. So we know when we're doing ENLs that there's no relationship between the structure of a leachable and the structure of your API. The leachable comes from the packaging material or the manufacturing component. So there's no reason for it to be similar to the API. Whereas in this case, for non-mutagenic impurities, they could well be very similar. If it's process related, could be very similar to your API. Having said that, the scientific approach, I would say, to doing read across is the same in both cases. The question you've got to ask is, can I find a similar enough chemical that has good data and use that to fill the data gap for my impurity? And I think it was encouraging that the EMA I think they've recognized that. So they were making reference to the draft ICQ3 guideline and some of the thresholds that are coming out of that project. But also all of the scientific effort. It's not like we can use stuff from other areas and apply it here because the science is the same. We're still doing the same comparison between my impurity and a chemical that's got good data. It just might be that in this case, if we can say the impurity is very much like the API, then we have data for the API. So that's a really powerful approach, I think.
I believe on this draft also it tells us how we need to, what are the approaches that we need to assess in order to do a read across. Because when you say a read across, it's just use data for a compound that you have data on, and then use it for your compound that you don't have data. But it's deeper than that. You need to assess multiple aspects in order to establish a good surrogate to use. So this version is better assessed on how we need to do that.
Yeah, to me, a good analogy of this is especially regarding structural alerts. So the analogy would be with nitrosamines. We can compare the structure of a nitrosamine with the structure of the API because they are very similar. They are highly similar. And so if you use a read across for this, you would end up with the same limit for the nitrosamine as the API, which is nonsense because we know that the nitrosamine is a potent mutagenic impurity and a potent carcinogen. So this kind of comparison is not adequate. So we cannot just look into chemical similarity. We have to look into several aspects before trying to establish surrogates. If not, we can end up in this kind of situation, in this nitrosamine situation that you are doing a terrible comparison. You are choosing a terrible surrogate. So it involves a lot of thought behind it.
Yeah, and even in read across, using in silico approach is good to avoid this kind of problem from happening. Yeah.
So you identify additional alerts, and if there's no additional alerts between the two compounds, then you can use them for a read across evaluation. Or maybe if your surrogate has more alerts than your impurity, then it could be used because you are being conservative in your evaluation, which would be...
Yeah, it was the first time that we said this word.
Yeah.
This word we like quite a lot.
We took a long time for mentioning toxicologists and conservative information.
Yeah.
So this kind of evaluation is this read-across evaluation is really interesting, and it's a really powerful tool that we have to establish a limit for the impurity and to evaluate if the amount that we are seeing in our stability studies is safe from a toxicological standpoint.
Mm-hmm.
So, and I would consider this to be
as a read-across.
A read-across alternative, but the reflection paper also mentions considering an impurity an API-like compound or a non-API-like compound.
Mm-hmm.
So I think this is really related to the read-across because you are doing a read-across of the impurity with the API, but again, you have to make some considerations because you can only do this kind of comparison if you know that the impurity doesn't have any structural alert that is additional in relation to the API.
Mm-hmm. Yeah.
If not, nitrosamines would not be a problem anymore.
Yeah, and I believe just like one thing that I want to add on these two topics is that MAA now tell us that we can also evaluate a read-across based on a qualitative approach also. So we don't need always to have like a point of departure to assess the acceptable intake for this compound. We also can do like a qualitative assessment, just like by saying, okay, that's fine. I know maybe this compound is similar to another, and based on the data that I have of the other compound, I know we can de-risk, as EMN say about it. So it's really, for me, it's get closer to what we do when we don't have quantitative data to work with. Now we make sure that we can also do that qualitative approach also. So, and that's why that's what API versus non-API like impurity is based on. It's a qualitative approach, just like by comparing the impurity as in the API and say, okay, so I don't expect any additional concern for the impurity because they are so similar to the API. So every effect that I already assessed for the compound, for the API, is like the same for the impurity as well. So this is a qualitative read-across for me. I see on that way. So yeah, I believe for API, just like one thing that is different from the draft version also is they try to limitate the changes that we can see on the API in comparison to the impurity, the changes in the impurity comparison with the API. So we can say that is a API-like impurity. So they want us just like to discuss that if the changes on the structure of the impurity is like very close to the API itself. So this is something that, because otherwise, what is similar, based on what data you can confirm that is similar.
We will use a Tanimoto coefficient, for example. How can you compare them?
Mm-hmm.
Yeah. So I would like to give a jump on the reflection paper, just to touch on a point that I think it's one of Martin's expertise.
Mm-hmm.
So I want to talk a little bit about the routes of exposure of the compound. So I don't know, what do we expect from different routes? Is this something relevant to be assessed on when we're trying to qualify an impurity?
Yeah, it definitely is. And it makes sense if you think about it. Like, depending on what the route of exposure, then there's going to be the chance for different types of toxicity to be observed. So you need to assess the relevant toxicological endpoints for the route of exposure. So I guess, I don't know, some examples. If you take in an oral drug, then that's going into the GI tract and being absorbed from there. So any systemic effects are therefore need to be considered. But you're less worried about things that might happen on the skin or in the lungs. I don't know, it depends on what you're doing. If you've got a topical drug, then obviously you need to start worrying about local effects as well at the site of administration. So yeah, I think it's a really key thing to know up front what you're doing, and then it impacts the strategy of how you assess that impurity.
And when we talk about topical compounds, what kind of assessments do you expect to do for them?
Well, the guideline or the reflection paper is very clear that sensitization is the effect that is considered most sensitive, the most likely to cause a problem. So yeah, potential to cause allergies is the big one. But yeah, luckily there's lots of science that's been done in that area that we can leverage to help us. Yeah, which is great.
Yeah, and on this topic, Nathan has a question for you, Martin, because yeah, I think this is a surprise for you guys because Nathan is not here, obviously, but he
wanted to be.
He really wanted to be, but he's not, and he recorded a question for you, Martin, regarding the topical aspects because. Here in the reflection paper, we can see that you were actually cited. Your paper was cited, obviously, regarding thermosensitization. And if Leandro can play for us.
Hello, guys. Olá, Ari. Tudo bom? Hello, Martin. How are you? It's a great pleasure to have you guys on the table with us and have you on this episode to talk about the reflection paper. Unfortunately, I will not be able to be on the table to make the interview and the talk with you guys, but I came here and I interrupt just a little bit this episode because I really want to make at least one of my questions about this topic. And then, Martin, how do you feel or seeing your publication about skin sensitization and talking about toxicological limits in the reflection paper? How do you feel about seeing your work in the publication?
Nathan, nice to see you, albeit a recording. And thanks for the question. It was really, I was just really proud, if I'm honest, to see the citation there. Like, this was a paper that we worked on at LASA a number of years ago. And I had no idea at the time that it would be of use in this field. So the dermal sensitization thresholds are an example of a TTC, a threshold of toxicological concern, and they came out of the personal care cosmetics sort of industries. That's where they were designed, and we've done a little bit of work in this paper to update them, working with the Research Institute for Fragrance Materials. So again, people who are interested in personal care cosmetics use cases. And I was probably the paper I'm most proud of that I've been a part of. So really glad to have been involved. But at the time, we had no idea that it would be useful for pharmaceutical impurities, that people would be able to pick it up and make use of those thresholds for non-mutagenic impurities and also for extractables and leachables. So to answer your question, Nathan, yeah, very proud to see it represented in the reflection paper. I'm really glad that it can be of use to people.
And one thing that I also think it's interesting to see, especially on the reflection paper, is that as we already seen on the ICH Q3E, we see that all the guides are being tied somehow. Mm-hmm. So here on the reflection paper, we can see that the limits established on the ICH Q3E, they are being used also. So they are presented here as they are presented in the guideline, in the ICH guideline. So it's really interesting to see how everything ties together and to see that although the chemical spaces are different from an ENL standpoint, from a nitrosamine standpoint, for example, we can see that the toxicological principles they are the same for everything. So I don't know if you guys agree with me, but to me at least, the reflection paper could be used for any impurity. It doesn't matter. I know the scope is for non-mutagenic impurity, but if you do this kind of evaluation for any other compound, it should be enough to guarantee that it's safe or not at an specified limit. I don't know if you all agree with me. So what are your thoughts on this?
They are so clear when they built that flow chart discussing the risk assessment for non-mutagenic, but totally makes it. Of course, we are not talking about here about mutagenic impurities because we already have guidance for this. But for non-mutagenic and other kind of compounds that could be, erased. So I think the best scenario, and I think EMA was so thoughtful and so smart to put this in a flow chart to be so easy to understand, to people to be familiar what they are expecting, not just because in Europe, but the rest of the world. I think it's another kind of document to harmonize with ICH and other big agencies around the world.
Yeah, and one thing just like that I found very interesting. First of all, I just want to thank Martin for your research, and also all the other researchers that were with you, not only on this paper that has established the new limits for sensitization, but the papers that come before that one, because for me it's quite relieved to have a limit to assess the toxicological aspect of impurity, and not just like by trying to find any discussion on the literature to try to assess the data. So for me it's really it's kind of a relief, really, that we started to have multiple TTC values for each to each toxicological aspect to consider, that makes us more harmonized in that way also. But one thing that I found very interesting about it is that the reflection paper is saying a lot about the limits from the ICH key 3E, but the limits are quite below the threshold that we need to qualify. So, you can use like 5 micrograms as an acceptable intake for this impurity, but I'm never going to use that, based on the maximal daily dosage of an API. It's really hard for us to need to assess the impurity safety on such a low limit. So for me, I found very interesting to have that. It's important to have, because now we can start to discuss based on that, but I don't know when we'll do that.
But I think the limits for the ICH Q3 are more they are established in the context of ENL.
For me that's perfect. We can use these limits for ENL. For me, it works very good. But when we're trying to qualify an impurity, then everything changes because the amounts are generally higher, and then we must use the other ways to qualify an impurity here. And they are higher because the limits of that regulations said to us that it's okay are higher also. So we only need to qualify an impurity if it crosses the qualification limit, but there's 2 other limits before it. That is the identification, the notification and the identification thresholds that are already higher than these limits that are being put here. So for me, I don't know. Doesn't make it's good to have it, but I don't know if we can use that discussion.
These limits probably won't be used for the qualification necessarily, but they are good to have, and assuming that an impurity is identified and it's below this general limits that we have, then it can be considered safe. So at least we are starting from a different point than just looking at the ICH M7 threshold. So it's good to already be on a higher level than the ICH M7, for example. And so, go ahead. Yeah.
Sorry, just wanted to add. I was just thinking, in ICH Q3 in the draft guideline, there's also this concept of class 1, 2, and 3 leachables, and it doesn't mention it here in the reflection paper. But just thinking, like, I would assume so. A class 3 leachable is one of low systemic toxicity concern, and that list is quite short at the moment. But in the future, that list may get longer. So I don't know. I guess there's the potential for crossover if you happen to have an impurity that was similar to something that has been described in ICH Q3. You might be able to leverage the work that's already been done. I guess it depends on chemical space, again, like we said earlier.
But anyway, it's incredible to have this kind of information and to start from a further point, right? So this ends up being quite valuable in the end for us.
Sure. One thing that I want to add about this discussion, like the correlation between multiple guidelines, is also about in the reflection paper we're discussing about the level of concern of the impurities that are being multiple aspects that need to be assessed. And it's quite similar to the structurable eligible as well, because we know that the route of administration for a structurable eligible is really important to be assessed, because we know that in the injectables form are like more concern other than others route of administration. And here in the reflection paper, we also have that. So during the whole evaluation of the toxicity data that we kind of may have for the impurity, we always need to make a comparison between the level of concern. So this is something that for me I find very useful, and it dialogues really good also with another guideline that is ICH S9, that also say to us that, okay, I know we're dealing with carcinogenity endpoint, so we know that maybe we don't need to assess mutagenicity of an impurity because we are already starting to, like the patient is already has cancer, and we are discussing about like these cancers that are really aggressive to the body. And we have a lot of aspect regarding what is called, once again, level of concern for this impurity, that for me it dialogues very well with multiple guidelines, but also during the evaluation of a non-mutagenic impurity as well, that we also need to be aware of the particularities of it, so we can assess with more more deeply what we need. Also, we need to get worried about. So this is another thing that is really
and especially considering these different concerns that the reflection paper presents, talking about daily dose, route of the administration, and the physical chemical properties, and so on. I really like this matrix that they presented, because I think it's a good tool to evaluate if you gotta be really concerned about the toxicological aspects of your impurity or not. But one thing that I miss here is how we could quantify this matrix. So, for example, we can maybe have an impurity in a drug that is used once in a lifetime, but the amount that is being qualified is quite high, would be over 5 milligrams, that it's on the higher concern. So we have one aspect of it being on the lower concern and another aspect of it in the higher concern side. So I don't know, how how would you deal with this kind of discrepancy? Would you go for the most conservative approach, or would you maybe make a mean of? You would try to find the average of the risk. I don't know.
Yeah, Marty.
Yeah,
You wanna?
I know.
You know.
Yeah. I know
I
that
don't know.
this is more.
Wait.
Go ahead.
No, we can both here can share our perspective because we assume the worst scenario. We always go to the worst scenario, more conservative, instead of to I don't know. It's more realistic to be conservative because we don't know what we're expecting to receive from the agencies as well. So being conservative is the best scenario. But I think if you have a good narrative to build in your full assessment, I think this can be changed by the end. So depend what kind of information do you have to build this narrative. I think this is the main advice here, because be conservative is the first scenario, but if you have a bunch of information, I think it's good to use it in your favor.
Yeah, and I agree. Like, I mean, you can always be conservative, and the way that the reflection paper is, or the workflow is constructed, if you consider the level of concern, you're trying to see if you need to continue or whether you can stop at that point, whether the level of concern is low enough to say, I'm happy, it's qualified. So conservatism is the safer way of doing it. But like Harry said, if you can build a good narrative there, you might be able to say, well, even though some of these are towards the higher side, overall the story says that this is low concern or moderate concern. But then that just has an impact on what you do next, whether you say we have enough evidence to stop, or whether you say no, we need to carry on and find more. Yeah, look for more data, I guess.
Yeah, I'm sorry for the philosophical question for this, but yeah, I think this is something that I see in all guides from ICH and other guides, and they present a risk matrix or how to evaluate the risk, but there are so many factors to include in this that it's difficult to quantify in the end what you have, because when we talk about a drug, we're talking about several different aspects of it. So you have a duration of treatment, you have a lot of things, and they don't converge necessarily. And so this is something that I miss on the guides, a more quantitative approach. So I don't know, maybe give a score 3 for a high concern thing, and maybe a score 1 for it, and then maybe a CPCA of risk kind of way of.
Yeah, but for
A
me,
matrix.
Yeah,
for
matrix.
me,
Yeah,
it's also good to, because we kind of like not having some like checklist endpoints to assess. So I believe AMA was really kind for us, not having like the bullets point that we must evaluate, you know. So for me, it's still consider the importance of having an expert review analysis. So for me, this is like where it stands us that us humans are still important to work with qualification, because if you could have some like a quantitative approach on that, maybe our use would be like less, you know.
yeah.
Because for example, in this guideline, it says about using others NUMS approach to qualify to assess the toxicity data of an impurity based, for example, in AI approaches. So we need to be careful about that we must have an assessor, like a human assessor on that matter also.
Yeah, definitely. I think this highlights the importance of the expert review of the information that we are presenting. And I think the risk-benefit considerations that we do to qualify an impurity, they are really a human aspect of it, and it's not something that can be quantified. That's true, because it really depends on a case-by-case scenario, and it's basically impossible to really give a guideline on this aspect. So I agree, we should be conservative on the risk classification that we do, but it's not something that we can really quantify and have just disregard a risk just because we consider that it's more towards the lower concern side instead of the higher concern side, and we always got to evaluate everything to make sure that it won't be a toxicological risk in the end in the product. And
I believe I have a final question. I want to know about you too. What do you expect for the future? Because as the guideline says about new approaches, methodologies, and as new approaches, methodologies are like everything that is not
in vivo, everything not in vivo.
I wonder what do you expect for the future if we are already studying something new? What go ahead.
Yeah, it's a really interesting question, particularly in the context of impurity assessments. I think I feel like everybody's talking about NAMs in the wider scientific community, and often it feels like people associate that with in vitro tests, which are a part of NAMs, obviously. But we're in an unusual situation here where it's not that straightforward maybe to generate in vitro data for impurities. So I think what I'm expecting to see more of in this space in the future is the computational side of things. And now that's already in the reflection paper, and we know there's lots going on, but I think we will just see more of that, more of those NAMs where we can use in silico tools like read across, like AAPs, like areas where we can take the evidence that we already have and interrogate it and use that to make a decision. So I think what I'm hoping for is more examples of this, more case studies, more maybe harmonization around those techniques that we've already been talking about today.
I think I think the same, because if we are all harmonized, this is the best scenario. But again, if you decide not to go further to in vivo tests and in vitro or even in silico AOPs, as Martin said before, if all the regulators start to accept more and more this information, because we have, again, the surrogate, the read across, we have so much information for 1,000 compounds that this can bring together and can bring and open the discussions, the dialogue better. This will be fantastic, and also more flexible, for post-approval in periods. So yes, I think more in silico, more computational instead of in vivo, because yes, the reflection paper just said start this before go to animals tests. But people still going to animals tests because there are some gaps, even when we are talking about new compounds, new development, about new APIs. But we already have so much database regarding other compounds, so much information about chemistry, structure chemistry, so we can use this in our favor. And I think this is my idea.
And one thing that I believe we are learning a lot with nitrosamine is that there's been some times that we kind of need to do an in vivo assessment or an in vitro also, but sharing information is really important. So other person, if it happens to see that impurity on this product, we don't actually need to assess once again, because someone already have done it. So for me, this is like the thing that we need to like work on, is that we need to share more information.
Yeah. And I know Nathan likes to quote Pasteur, but I prefer to quote Newton. So in this situation, we are really standing on the shoulders of giants. And we can see better because we have a lot of information, and we don't need any, well, actually, we might need, but we usually don't need in vivo data to evaluate the toxicity, because we have so much information already that we already know. We know different pathways. We can establish different correlations, and from this we can make pretty safe assessments and pretty comprehensive assessments to evaluate the toxicity of an impurity without really needing to evaluate it on a little mouse or something like this. Yeah. And it really makes the development of new drugs faster, and it also contributes to the safety of the population that is receiving this drug or this medicinal product. So, yeah. So I'm really thankful.
Yeah, no, I believe
Yeah.
we can go. Yeah, for sure.
So I'm really thankful to have both you guys. I think these kind of discussions are really helpful for us, and we can definitely see some perspectives on the qualification of impurities, and we can understand better how we can do this and how this is important for the industry and important for the patients also that will end up with these products, and we can guarantee safety in all process. So I'm really thankful for you both, and we really hope to have you again sometime in the future.
Yeah, for sure.
Hopefully in person.
In person.
Yeah.
Better.
Maybe we can talk to Nathan and Maria. So all these talks should be done in person.
we need to travel.
Oh, you're going to Brazil. Oh, you're going to here. Come here.
No, let's go to Japan. We can do this kind of talks in Japan or any other beautiful country in the world. So I'll let Nathan know.
Yeah. Oh, that's perfect.
Thank you for having us. It was really amazing to have this conversation again. I think always about qualification impurities is one of the topics that I love to discuss. I was there once, and it's nice to be here again on the other side. So now we are all the same as well, but it's good to see you again.
It was incredible.
Yeah, same. Thank you. Obrigado.
Nada.
Well,
Obrigado, Martin.
obrigada, Martin.
Foi incrível.
That was really good. Thank you.